right. So the lab that's due this week was completely online, and we ended up not meeting this week at all, either online or in person. And it'll be like that for next week too. So no worries about that. 1 second. Let me look at something. So make sure I'm telling you the right thing here. Classwork lab. So the ecological footprint lab. Yeah, it's the ecological footprint lab. So yeah, that's completely online. You shouldn't. I mean, if you need help, let me know. Which is a good time to remind you for these announcements. I will be traveling this weekend. So if I'm a little slow to reply or respond, I apologize in advance. What else for announcements? Okay, speaking of lab, I've been hinting around for actually a long time now. I keep saying, I'm dragging my feet on grading the lab reports because the longer I wait, I have no way of knowing when you did it. As long as you did it before I look at it, then you're good, right? So because of that, I dragged my feet. But as of yesterday, I started not dragging my feet. So you'll start seeing some grades soon. So really, I mean, if you haven't done it and I haven't already given you a 0, because I did start it yesterday, and if I haven't already given you a 0, then you're really, really running out of time. So any questions about that? All right. So again, next week's lab will be online. Monday's lecture will also be online. Today's attendance will be sort of like we've been used to be doing it. It'll be keywords. So for those of you live online, just make sure you email me before 9 a.m. with those keywords. Anybody watching the video for attendance, you also will just do the keywords. So there won't even be any questions this Friday. You can have a nice weekend. But if you're watching the video, not if you're live in person, but if you're watching the video and you send me the keywords, also write at least 3 sentences about 3 separate things I talked about, just as proof that you watched the video. And that's it. So any questions before we get started? All right, I'm going to get into it quickly. We are behind and down to crunch time. All right, so I know people can hear me. We've confirmed that, but can someone confirm that you can see the regular old PowerPoint?
Yeah.
Perfect. Thank you. All right, so the last term we ended with was gene pool. And the way, obviously in front of you, you can see how the book describes it. But my description was, think of all the genes in your body. That's called, in your cells, right? That's your genome. So another way of thinking of a gene pool would be all the genomes, right? So the genome of every individual that's in a population. So now that we know what a gene pool is, we can move into this next concept, which is analyzing gene pools. And you've already done this in lab. And analyze will be the first attendance keyword. Analyze, like you see right here. Of course, that says analyzing, but analyze is the word. And if someone sends analyze, they're not going to lose points. That'll be just fine. Analyzing, it doesn't matter. Anyway, picture, if you will, a bunch of flowers. And we've talked about flowers a lot, especially in the genetics, right? Actually, you know what? I should change this. Forget those flowers. Well, I'll stick with it because that's what I have the visualization for. But you could also picture the fish that you dealt with. So we're going to go with this wildflower population that are either white or red, but we'll come back to the fish idea later. So here we go. We have this wildflower population. Half of them, or no, I shouldn't say half. A portion of them are white, a portion of them are red. The allele for the red flower is dominant to the allele for the white flower. So big R is for red, little r is for white. Therefore, really quickly, even though this isn't written down as a reminder, and some of you judging by the exam need this reminder, homozygous dominant, big R, that would be a red flower. Heterozygous, big R, that would be a red flower. Homozygous recessive, little r, that would be a white flower. Anyway, now that we know that, let's talk about analyzing these gene pools. The genotype frequencies can be calculated from the allele frequencies, which is a book way of saying we can figure out the percentages of the genotypes by using the percentages of the alleles, right? And again, so when you hear the word frequencies, that's what the word the book uses, but basically that means percentages in a sense, except with frequencies, it's expressed as a decimal instead of an actual percentage. So for example, you might have the 0.5, no, I'll do something different, 0.6 frequency of big R, which would mean by definition, you would have to have 0.4, right? Because 0.4 plus 0.6, basically we're saying 60% and 40%, right? It all has to add up to 100%. So that's what the Hardy-Weinberg formula is. You've already done the lab, but now we can actually talk about it a little bit more. And I would assume you watched the videos because it said you had to watch the video embedded in the lab. But we talked about this, or I talked about this in that video. It's basically all it is. It's just a way to quantify, right? To quantify the genotypes. Like, okay, well, we have, I don't know, 50% homozygous dominant. We have 25% homozygous recessive. And we have 25% heterozygous, right? It's just a way to calculate it. And the formula, there's actually two formulas, but the main one is the one that's actually called the Hardy-Weinberg formula is this. P squared plus 2PQ plus Q squared equals 1. And that really can be a little intimidating if you're not into math. So what you could think of it is this. Yes, there's we have terms that we could move around algebraically, but we don't do much of that in this class. So you can just think of these as genotypes. So we have homozygous dominant. Oh, that's so horrible. We have heterozygous, and then we have homozygous recessive. And again, I just gave you an example, but I'll give you another one. If we had, I don't know, 60% or 0.6, if we're doing frequencies, if 0.6 was P squared, and again, you could also think of that as 60%, and the heterozygotes were, I don't know, 20% or 0.2. There we go. I know that looks so horrible. I apologize. Think about it. If it's a percentage, let's start with percentages. People know this better. If we're looking at this population and maybe, okay, we know that 60% are heterozygous dominant, 20% are 60% are homozygous dominant, 20% are heterozygous. What must the percentage be for the rest of them? Because these two, 60 plus 20 equals what, guys? 60 plus 20. I know we don't. Yeah, there you go. I was about to say, I know we don't have any math majors, but there we go. So those two equal 80%. So what must this percentage be for the homozygous recessive? We're talking about percentages here, so it all has to add up to 100%. We're saying this population is 80% these two.
20.
Thank you. All right, so for the person that said 80, who was that?
Joanne.
Yeah. All right, Joanne. When you email me for your attendance, remind me that you got that so you get a little extra credit. And Schmidt, was that you with the 20?
No, that wasn't me.
That was me.
Well, whoever me is, when you send me in your attendance words, remind me that you got that. So a little extra credit for your math skills this morning. But yeah, that's basically that. And that's, if you get any questions on the exam, that's what it's going to be. The most easy one is going to be just know that they all have to add up to 100%. Or in this case, like I said, if we're writing it as frequencies, then instead of saying 60%, it's 0.6. Instead of saying 20%, it's 0.2. So again, in this one, instead of saying 20%, it's 0.2. But you won't mess it up because it's a multiple choice exam. So it's not like you'll think, oh, do I need to do it in percentages or decimals? Because I'm only going to give you one choice, right? Your choices will either be in percentage or decimals. Anyway, that's what the Hardy-Weinberg formula is. It's a way of quantifying not only the alleles, but more relevant to at least this discussion, the genotypes, right? That being said, the easier one in my opinion, is like the allele frequencies. There we go. The allele frequency formula. And it's basically the same concept, except remember, with phenotypes, like at the bottom, there's 3 different phenotypes, right? But with alleles, there's only 2. There's either dominant or recessive. So this is the real easy one. So for example, if 60% of your gene pool, oh, where'd my thing go? There we go. If 60% is dominant, then what must be the percentage of recessive? Anybody who hasn't guessed yet? If 60% is dominant, then what must be the recessive percent?
40.
Good. Who said that?
Joanne.
You're on. All right, you're on a roll. So yeah, there you go. 60 plus 40 equals 100, right? Or 0.6 plus 0.4 equals 1. It's that simple. Now, granted, I am going to give you one more. I'm going to give you a complicated version a little bit later, but that complicated version probably won't be on the exam. But anyway, you need to know that, and you definitely need to know there'll definitely be a question where it just shows you this bottom formula, and you say, "What is p?" And it asks, "What does p squared represent?" Or "What does 2pq represent?" Or "What does q squared represent?" And the answer is those things right there, right? Dominant, homozygous dominant, heterozygous, homozygous recessive. And again, I will show you, we'll get a little bit more complicated a little bit later when we actually do use a little bit of algebra and we take square roots of things. But for now, it's a pretty simple concept. So any questions so far? All right. Let's talk about population genetics and health science. So in other words, what I'm about to do is show you why this is even useful for people who aren't biologists, right? And make sure it makes sense. If you're out there studying, you're a field biologist and you're studying a population of endangered wildflowers and you need to know this and you need to know that about them, sure. But what about the rest of us? Why would that matter? So here we go. Bear with me. This is just an example. I'm definitely not going to ask you anything about this. You might have one question where I ask you to solve for a term, like we use algebra to use Hardy-Weinberg, but most likely not. All right, so I'm going to go through this. If it doesn't make sense, let me know. I'll briefly try to help you. If it still doesn't make sense, we'll have to meet online because we're behind times or behind schedule. But again, if you don't get it at all, don't let it stress you out. That's only one question. If anything, all right, before I even get started, if anything, just know that the big picture here is this is why we use the Hardy-Weinberg formula. This is how algebra is used to do something useful. So there's a disease, PKU. Not that you need to know this, but again, just a little background. PKU is the inability to break down the amino acid phenylalanine. And remember, from way back when we talked about cellular respiration, we kept talking about breaking down glucose, but then at the end, we're like, hey, but by the way, you can also break down other things. And one of those things was amino acids, right? You need to be able to break down amino acids and digest them for their energy and for their parts. Anyway, if you have PKU, you can't break down that particular amino acid. It's not a good thing. I don't have time to get into why, but you can look it up if you want for independent work. What happens if you have PKU? Anyway, this disorder is caused by a recessive allele. So to remind you, I'm just going to use a Q here. And I don't know what the letter is, but if you had it, you would have to have little q, right? You would have to be homozygous recessive to have it. You couldn't be heterozygous because it's recessive, right? So if you were heterozygous, like if you were big Q, little q, you'd be completely fine because you have at least one dominant allele. So you don't have the recessive condition. And obviously, if you were big Q, you certainly wouldn't have it because you don't even have the recessive allele, not even one. Anyway, that being said, what we know already, right? So if you're into public health and you're trying to figure things out, and you could easily figure out from hospital records that 1 out of 10,000 babies in the U.S. is born with it, right? 1 out of 10,000. So now we have a number. So again, I wish I had this up here, but Q squared plus 2PQ, sorry for those lines, plus little q squared. Oh, that's right. Well, I should put that as a p squared. But in this case, It's almost the same thing. Anyway, p squared plus 2pq plus q squared equals 1, right? That's the formula we're dealing with. And what do we know? We know 1 in 10,000 babies is born with it. And another way of saying this bullet point right here, that means 1 out of 10,000 babies has the genotype q squared, right? So of this equation, this is given. We know that q squared equals 1 out of 10,000. Now, if you were to express that as a decimal point, like 1 out of 10,000 is the same as saying 0.0001, right? So this is the frequency of homozygous recessive in our population. So q squared equals this number. But what we want to do is figure out the other genotypes. So what we need to do then is solve for q. So if q squared equals this, I don't know how many of you are good with algebra. If you do it on one side, you have to do it on the other. So let's get rid of that squared, right? So we take the square root of q squared, we take the square root of 0.0001, and this is what we get. Q equals 0.01. Now we want to know what P is. Oh, we know what Q is. Let's figure out what P is. So remember, Q plus P equals 1, right? So this is what this is the formula we're dealing with now. Q plus P equals 1. Sorry, that looks like garbage. So, all right, if Q plus P equals 1, we're trying to solve for P, then we do, again, algebraically, we're going to subtract Q from both sides. So then you're just left with P equals 1 minus Q. And that's where this comes in right here, right? Same thing. You can switch it around the equal sign. So 1 minus Q equals P. So again, we know we're trying to solve for P. We know what Q is because we just solved it right here. So we just plug it in. So 1 minus 0.01 equals P. So in other words, 1 minus 0.01 equals 0.99. That means P equals 0.99. Now, there we go. Now, by knowing how many babies were born in the U.S. with this disease, we now know right here the percentage, we know the allele frequency for the recessive. We know the allele frequency for the dominant. We already figured out Q squared because that's the information that was given us at the beginning. And now because of that, we can figure out P squared and 2PQ. I don't know why, but for some reason I only wrote down 2PQ. But now we know the percentage of people who are heterozygous. Oh, yeah. And now I remember. The reason I wrote that one down instead of P squared, because, again, we can also calculate P squared. But this is important because, again, it's a recessive allele. So if you're into public health, not only do you want to know how many people have it, but more importantly, it's helpful to know how many people are carrying it, right? Because people who are carrying this disease might not even know they have that allele because they don't have the disease. They just have the allele. So it helps for public health to know, all right, well, we know about 2% of people are carrying that allele. So anyway, that's why it's useful. You might have one question where I give you something like this and you have to use a little bit of algebra to figure something out. But if I do, it'll be easier than this. And again, if I do, it'll only be the one question. So if that's stressing you out, don't let it. Just roll with it. Just know that that's one of the ways that Hardy-Weinberg is useful in just everyday life, right? It's good for public health. You can figure things out like that. Anyway, now let's jump back into, while we're talking about Hardy-Weinberg, we're going to talk about microevolution as a change in a gene pool. So this is the stuff that we were talking about for your lab, the goldfish, the second goldfish lab, right? When you're trying to decide, where we're going to have, where the percentage is going to change of orange fish, white fish, and the orange with white spots, right? You were looking to see how that changed. So let's jump into this right now. Actually, before I even jump into that, what I want to say about microevolution, and you probably already know this if you did the lab because you watched the video, but when most people think of evolution, they probably think of something like, oh, humans came from monkeys, which is not an accurate statement, but it's a lot of times what people think, right? When people think of evolution, they're thinking the grand scheme of things, like the fact that everything has a common ancestor. And sometimes that doesn't sit easy with people. But another type of evolution is microevolution. And that's just smaller changes. That's just changes in the gene pool. Like, I'm just making this number up, but let's just say, I don't know, 15% of the human population had blue eyes 100 years ago. And let's say maybe 17% of the human population has blue eyes now, right? That would be microevolution. Like, we're not changing into a new species. It's just slight change of the number of the percentages of people with blue eyes. So here we go. Let's talk back. Let's jump back into what the book says about this. A non-evolving population is in what is called a genetic equilibrium, also known as a Hardy-Weinberg equilibrium. And you do need to know that for the exam. Not only do you need to know the Hardy-Weinberg equation, but you need to know what the Hardy-Weinberg equilibrium is. And before I read what the book says, the Cliff Notes version, the short version is Hardy-Weinberg equilibrium is when evolution's not happening. It's when nothing changes, right? And again, I'm not talking about big evolution, like, oh, this species changed into that species. I'm talking about the tiniest of evolution, right? Like the percentages of orange fish doesn't change. It's constant, and the percentages of white fish and the percentages of spotted fish does not change, right? That's Hardy-Weinberg equilibrium. What your book says about it is when the gene pool remains constant from generation to generation, right? So it's the same thing I said, just in a more academic way for you. Another way of saying that is the frequencies of alleles, which are P's and your Q's, right? Your dominants and your recessive alleles, and the percentages of genotypes. So your homozygous dominants right there, and your heterozygotes right there, and your homozygous recessive right there. They don't change. So again, these two bullet points are just a long-winded academic way of saying the population is not evolving. So because generation to generation change in the allele frequency is evolution at the smallest scale, it is referred to as microevolution. So sorry, I jumped into that. These two bullet points are very separate ideas, right? The first bullet point is introducing you to the idea of what Hardy-Weinberg equilibrium is. And it's really an academic idea. It's more of a hypothetical thing. Like, things are constantly evolving, even if we can't see it, right? There's always going to be alleles changing. Anyway, so that's what that bullet point is. And then on a completely separate note, your book is just introducing what I've already introduced you to, which is the term microevolution. So again, microevolution, it's not species changing from one species to the next, right? We're just looking at changes in alleles, changes in genotypes, changes in phenotypes, things like that. Any questions so far? Okay. And that's really always, well, sorry, excuse me. With that being said, let's look at the mechanisms of evolution. And I want to remind you as I've been saying throughout this chapter, because so far we've been talking about natural selection. Like, that's almost all we've been talking about. We talked a little bit about artificial selection, but now we're going to talk about things other than natural selection. So as I've been saying throughout this chapter, natural selection is just one of the way things evolve, right? There's other ways things evolve. So let me say this too before we jump into this. Hopefully I'll make it quick because I know we're short on time. But somebody asked a question, and I don't know if that was supposed to like a suggestion, like, hey, this would be a good quiz question, or if they were literally asking me. But I said on Wednesday that one of the things that is needed for natural selection to occur is an overproduction of offspring. And I know when I was a young student, that kind of confused me. Like, I didn't quite understand why. So really quickly, think of it like this. Imagine there's like this beautiful tropical island way out in the middle of the Pacific. And this is not a very realistic example, but it should be easy to follow. Anyway, big, beautiful island, nothing but plants. There's no animals because it's just too far away for any animals to get to. And then out of some freak accident, maybe like a bunch of birds that were getting transported on some ship to go to some zoo in some country. That ship sinks, those birds escape, and somehow they make it to that island, right? So now you have this new population on an island. And it just so happens that island is perfect for those birds, right? It's this exact same temperature that they like. It has the exact food that they like. There's obviously no predators. There's no diseases. There's no parasites. There's plenty of places for them to nest, right? So life is perfect for them on that island. So what would happen over, let's just say it takes thousands of generations, right? Thousands of generations, they just keep reproducing. And there is no evolution. There is no natural selection, right? Because there is no need for them to change. Everything is perfect. There's no advantage to being faster. There's no advantage to being slower. There's no advantage to having a longer beak. There's no advantage to having a shorter beak. There's no advantage to, having harder eggs, or no advantage to having softer eggs, right? There's no advantage for anything because everything is perfect. So nothing changes. And then one day, eventually, because remember, they just got to that island. There's nothing else living there, and they're reproducing. Eventually, they get to the point where they have hit the level where it's no longer sustainable. They are now eating more food than can be produced by the plants that they eat, right? So now all of a sudden, nothing's changed except for the fact that they now have an overabundance of offspring, right? They're now more individuals are being created than there is food to feed them, right? So now all of a sudden, some of them are starving to death. So now all of a sudden, advantages come into play. So maybe it is advantageous to be fast. So now the population starts changing, right? Because the faster ones are the ones that are able to fly from food source to food source, and they're eating first, and that leaves the other ones to die off. So now over generations and generations, that population is going to become faster, right? And that would be natural selection, right? Because prior to that, there was no overabundance of offspring. Therefore, there was no competition for any resources. Therefore, there was no change. There was no natural selection, right? Because everything was perfect. Okay. Does that make sense? Does anybody have any questions about that throwback about this idea of you must have an overpopulation for natural selection to occur? Okay. And again, to be clear, really what that's saying when we say you must have an overpopulation, you should really take it to the next step, because what that leads to is competition for resources, right? You have to have competition for resources, whether it be food or shelter or water, whatever it is you need. You need competition. That's what drives natural selection. And on that note, let's say this. Natural selection, again, is not the only way you can evolve, right? Or not the only way that populations evolve. Because think about it. There's still mutations. So that population of birds I was just describing, yeah, some of them are going to be born a little bit taller. Some of them will be born a little bit shorter. Some will be born a little bit faster, a little bit slower. Some will be born with, thicker beaks or shorter beaks or different colors, right? Yes, they would be born that way, but until there was this overpopulation, there would be no advantage to that. So yeah, there might now be a small percentage of birds that have a longer beak, right? But because there's no advantage, the percentages, they're not going to change a lot, but they could change by chance, right? So let's think about what I just said and talk about this stuff here. What mechanisms can change a gene pool? We already know about natural selection, right? Natural selection is the big one. That's actually what promotes adaptation, right? So with natural selection, you're getting the best of the best, right? So natural selection, in my analogy of the birds, being fast was an advantage. So eventually they became fast. Being fast was an adaptation to that environment. But there are other ways that things change, and we will talk about this. The first one is genetic drift. Genetic drift is a change in the gene pool or in genes, whatever, due to chance. If you don't know anything else about genetic drift for the exam, know that. So we're talking about changes in the gene pool here, right? We're talking about evolution. But now it's not natural selection, because that wasn't by chance, right? The example I gave, it wasn't by chance that the fast birds were surviving. They were surviving because they were better at getting food. So that was natural selection. However, prior to that, when there was only a few birds on the island and there was no competition, like I said earlier, there could have been, there's already variation, right? There's variation in the population. Some of them are already faster than others. Some of them are already slower than others. But since they were living in a paradise and they had all the food they wanted and they had no predators to escape from, there was no advantage to being faster or slower. So that being said, let's use your book example. Your book example is this population of wildflowers, right? They're red and they're white. Those are the two phenotypes. The genotypes are homozygous dominant, homozygous recessive, and of course the heterozygous right here. Now, in this population, wherever they are in this environment, there is no advantage to being red, and there is no advantage to being white. So by chance, when this generation mates, these ones with the squares on them, those are the ones that mate, right? So by chance, this one, and these two, those are the ones that share pollen, right? The rest of them, these right here, they didn't mate. It's not because one was better than the other. It's just by chance, right? So anyway, this population, when we first look at it, it's 70% of the dominant allele and 30% of the recessive allele. Not because one's better than the other. That's just the way it was. So again, those things mate. They have some offspring. We look at the second generation. Now, all of a sudden, by chance, because of the ones that made it, and we could do a Punnett square for all those, and I could show you how, but let's not do that. By chance, the ones that made it, these are the ones that made it. So the second generation now, it's 50-50, right? It's 50% dominant allele, 50% recessive allele, or 50% red, 50% white, however you want to look at it. And that's not because red or white's better, right? That's just by chance, that's what happened. And then by chance, these two are the only flowers to mate in those generations. So then the next generation, it's all red, right? The next generation, the third generation, we have completely bred out the white allele, right? The recessive allele. Not because red's better, just because that's just what happened, right? So you could do these three boxes and rewrite this any way you want, right? Where the percentages change any way you want. You can make it where the red go extinct instead of the white, because it's all by chance. There's nothing driving that. Does that make sense to everybody? Or does that, a better question, does that not make sense to anybody? All right, so remember, again, genetic drift is all by chance. And I've thought of a way to try to remember it, but I really can't. The stupidest thing I can come up with, well, the only thing I can come up with is just stupid. There's this movie called Fast and the Furious Tokyo Drift, and it's all about them like racing or whatever. I haven't seen it. But whatever it is that they're doing, other than stupid, it also looks risky. So it looks like they're out there taking a chance, acting the way they're acting in their automobiles, right? So anyway, that's the stupid way, the only way I can think of to help you remember that genetic drift is changes by chance. Now, here's where it gets slightly more complicated, but not too complicated as long as you're paying attention. Under this term, genetic drift, we have two subcategories, right? So the next thing I'm going to tell you, the next two things I'm going to tell you, but not the bottleneck effect and founder effect, those are both a type of genetic drift. They're just a little bit more specific scenarios. The bottleneck effect, that'll be the next keyword, bottle. Bottle is the next keyword. Anyway, the bottleneck effect is an example of genetic drift, and it results from a drastic reduction in population size. So that picture I just showed you earlier, the genetic drift was, hey, these flowers happen to mate with those flowers, and those flowers happen to mate with these flowers, right? So in that case, the chance that was happening was what mated with what. In this case, we're just talking about a reduction in population size, but it has to be by chance, right? So for example, if a bunch of humans died from COVID-19, like really, like the bigger numbers than we had now, that would not be the bottleneck effect, because that wouldn't be by chance. That would be, let's just assume every human got exposed to it for the most part, but only a chunk of them died. That would be natural selection, right? Because some people were more genetically predisposed to die from it than others. But in bottleneck effect, you lose a big portion of the population, but it is completely random. It's completely by chance. It has nothing to do with one being having an advantage in the other, right? So again, another example would be the peppered moths. You could almost call that a bottleneck effect for the white colored moths, but that was not genetic drift. That was not by chance. That was natural selection, because there was an advantage to being dark and a disadvantage to being light. So anyway, with that being said, let's keep going. The reason they call it a bottleneck effect is because basically the population is kind of passing through a bottleneck. And if you don't know what I mean by bottleneck, think about driving on 64 West and you hit nitro, right before the bridge, right? You go from like 3 or 4 lanes and then it squeezes down to 2, especially now where they're doing construction, right? So when you're going, that you're just passing through a bottleneck. And I'll show you a picture a little bit later, and hopefully that'll make more sense. But yeah, that's sort of what we mean by bottleneck. Again, you're just really reducing the population size. Anyway, what happens when you go through a bottleneck? Well, it decreases the genetic variability, right? So that's not a good thing, and we'll talk about that in later chapters, why genetic variability is a really good thing. But when you go through a bottleneck effect, you lose some alleles, and by chance too. So you might lose some really good alleles. And it also results in a loss of individual variation. And again, without variation, you can't have natural selection, which means you can't have adaptability, right? Meaning you can't have an organism, or excuse me, a population that's a good fit for its environment. Here's what we mean. This is a good way to visualize bottleneck. So here's the original population, and imagine we're just looking at the gene pool, right? There's these different alleles. There's a green allele and an orange allele and a purple allele. And now you're going to severely reduce the population. And it's all going to be by chance, right? There's no advantage to being red, purple or green or orange. So you dump out the bottle, and by chance, not because one color was better than the other or smaller than the other or smoother than the other, right? They're all exactly the same, just a different color. And this is what you're left with. Now your new population, not only is it much smaller, but it's almost entirely purple, right? You almost got rid of all the greens and you did get rid of the oranges. And that's what a bottleneck effect is, right? It's by chance, and the result is usually, are obviously a lot less genetic variability, which is a bad thing. And we'll talk why later. Your book gives 2 real-life examples of when this has happened. Cheetahs have done it twice, at least 2 genetic bottlenecks. The first one was about 10,000 years ago at the last ice age. And then the most recent one was from humans. It was human-caused in the 1800s, where farmers hunted them to near extinction because, the cheetahs were eating their livestock. Anyway, that's just an example. I'm putting next to that because I won't ask you any questions about the example. There are questions on the study guide, but they won't be on the exam. Anyway, with such little variability in the gene pool, these cheetahs have reduced their capacity to adapt to environmental challenges. So again, they had all these different alleles in their gene pools, and they had all this variation, but now they don't. So, if they get hit with a cheetah COVID, right? If they get hit with some kind of bad disease, and if it could kill one of them, it might be able to kill all of them because they don't have that much genetic variation. But unlike humans, think about, again, COVID's just the most recent example. Some people, it killed some people, and some people didn't even know they had it. And a lot of that had to do with their genes. We don't know how much yet because, people are still trying to figure it out. But a lot of it had to do with genetic variability. Anyway, any questions so far? So again, big picture, we're talking about evolution other than natural selection. Particularly, we're talking about genetic drift, which is changes just due to chance. And specifically, we're talking about the subcategory of genetic drift. We're talking about bottleneck effect, which is a huge reduction in the population. So that being said, let's look at the next type of genetic drift, which is called the founder effect. And again, this is all by chance. And in a sense, it's a lot like a bottleneck effect, except the mechanisms that get you there. The founder effect is when few individuals colonize an isolated habitat. Basically, it represents genetic drift in a new colony. Before I even talk about this next bullet point. So in a sense, like I said, it is a lot like a bottleneck effect because in a sense, you're going from a large population to a small population. The only difference here is instead of you losing like a large amount of the population dying, instead of that, what's happened is a small portion of the main population goes somewhere else. So nothing dies in the founder effect, but the result is the same. The result is a new small population that does not have much genetic variability, right? So you eliminated the larger portion of the population, the original population, except in this case, it's not by death. In this case, it's because a small portion of the original population went somewhere else. Anyway, the founder effect can explain the relatively high frequencies of some inherited disorders in some small human populations. So, for example, there's a small island in the Pacific called Pingalap, and you can click this and watch a short video about it. I think it's interesting. Anyway, they have more colorblind people than any place on Earth because among the few people that colonize that island, there just happened to be a higher percentage of colorblind people there than there were in the regular human population. Then the Amish, they have quite a bit. They have quite a few different ailments that are higher in percentages in their population than there are in others. And I like the Amish example because of this. Your book says represents genetic drift in a new colony. And a lot of times people accidentally say a new island. They'll say it's an island. And oftentimes it is an island, like Pingalap and Martha's Vineyard. There are very often islands. But sometimes the new colony, this new isolated colony, It doesn't have to necessarily be an island. The Amish are not on an island. They're not physically isolated from anybody, but they are culturally isolated. So because of that, there's not a lot of gene swapping, right? They've got their gene pool, and there's not a lot of genes leaving, and there's not a lot of genes coming in. So anyway, the other example is Martha's Vineyard. This is actually where American Sign Language was invented, because there's a higher population of deaf people in Martha's Vineyard than there is in the general human population. Because, again, when the first humans got to Martha's Vineyard to settle it, or at least the first Caucasians, there was a higher percentage of deaf people. And I'm not saying there's a bunch of deaf people. I'm just saying there was, I don't know what the numbers are, but it was higher. Let's say, 5% of humans are deaf. It could have just been that 6% of the humans on Martha's Vineyard that originally went there were deaf. And eventually, because of that small change at the beginning, that you had a larger change at the end. Again, I just made those numbers up. But anyway, for the exam, you don't need to know those examples. But, maybe I'll ask them extra credits questions about them, but you don't need to know. So any questions about the founder effect? All right. Here's the easiest one, in my opinion. And I wish your book would have presented it a different way. I think gene flow should have been discussed first, because I almost found myself using it on this slide. Because I was talking about the Amish, right? I said that, yeah, they're culturally isolated, and they're not exactly, mating with other people. So there's not a lot of genes coming in, not a lot of genes coming out. And what I mean by that, the word for that is gene flow. Gene flow is another source of evolutionary change, and it is the genetic exchange with another population. So it's basically just mating. And it's hard to think about that with humans, because humans are basically, we can just go essentially wherever we want on Earth. And there is no population, for the most part, that is isolated from another. So just think of something other than humans. And again, think about those birds I was originally talking about earlier today. Let's say they're on their island. They're perfectly adapted to it. And let's say there's two versions of them. There's light colored and there's dark colored, right? And they're about 50-50, because there's no advantage to being light and there's no advantage to being dark. But then all of a sudden, here comes another round of birds. Somehow the same, some more birds of the same species made it to the island, and they're all dark, right? You get 5 more dark birds come in. And now, just by chance, well, I don't want to use the word chance because I don't want to confuse anything, but now these birds have made it to the island, and they brought in some dark alleles. So now all of a sudden you have more dark birds than you have light birds. Not because being dark is better or being light is worse. It just so happens the ones that made it to the island were dark. And that is gene flow. Gene flow is interesting because it may result in a gain or loss of alleles, right? So you might make things more diverse. You might make things less diverse, depending on what makes it, what breeds. But one of the things it definitely does is reduces genetic differences between populations. So again, those dark birds arrived to our little magical island, this fantasy island I'm talking about, right? The dark birds arrived from there. So now you have more dark birds on that island. And now the island that they left now has obviously fewer light dark birds because they left there, right? So now whatever differences those two islands had of birds, now they're a little bit more evened out because of that. Anyway, if you download the PowerPoint, you can click this and watch another short evolution video. This one talks about gene flow in it. So any questions about gene flow? It's a very simple concept. We're just talking about genes flowing from one population to the next. All right. So those are the big ones, basically. If we're talking about mechanisms of evolution other than natural selection, those are the big ones. But then now we're going to go back into natural selection, because there were some things we didn't necessarily look at when we talked about natural selection. So let's do it. With those other things out of the way and in mind, we can again say that it is only natural selection that leads to adaptive evolution. So remember, adaptive evolution means it's not by chance. Adaptive evolution means you're making the population a better fit for its environment. And only natural selection does that. And again, natural selection is evolution that results in a better fit between organisms and their environment. Your book gives this example of the blue-footed bluebee, which is a type of bird. It's an excellent example. Its body and build are streamlined like a torpedo, which is perfect because the way it hunts is it kind of glides around up in the air over the sea. Then when it sees a fish that it wants, it kind of dives in, right? So the quicker it goes, the better. The easier it can glide through the water, the better, right? Because that's how it gives its food. It also has a very large tail that functions as a brake. And also the females prefer males with bright blue feet. We'll come back to this concept in a little bit. But yeah, so natural selection made this, right? So again, using assume these are the birds I was talking about in my little hypothetical situation earlier. There was no advantage being taller, shorter, quicker, slower, whatever. But now all of a sudden, because of overabundance of organisms and things starving to death, well, the better built you are for catching fish, the more likely you are to survive. So in this case, the fatter ones were less likely to eat. And the skinny ones were more likely to eat. Therefore, the more streamlined ones were more likely to survive and then therefore mate and then pass along their genes. So therefore, eventually, the population became more like that. And now the population is better built to live in that environment. And obviously, I could say the same about these bullet points, but I'm trying to save time. Anyway, the blue-footed booby, just an example. There won't be any questions about that on the exam, but there are in the study guide. If you download the PowerPoint, you can click here and watch a video, yet another short video about natural selection. I would ask if you have any questions, but we're not done talking about this particular portion yet, so let's keep moving forward. There's a blue-footed booby. That's what it looks like. You can see why they call it a blue-footed booby. Anyway, first thing we're talking about here is evolutionary fitness. When we're talking about a closer look at natural selection, you need to know what relative fitness is. It's the contribution an individual makes to a gene pool relative to the contributions of other individuals. In other words, it's saying it is survival of the fittest. That's a slightly inaccurate way to say it, but I'm sure you guys have all heard it. It's survival of the fittest. And the reason why survival of the fittest is a slightly inaccurate term is because that's only part of the equation, right? You definitely need to be able to survive, and if we're talking about natural selection, but if you're not passing along those genes, whatever genes it was that made you survive, if you're not passing them along, then that's not natural selection, right? Yeah, good job, you survived, but if you didn't pass along those genes, that's not natural selection. Or, I don't know, if you're just, I don't have time to talk about it, but yeah, there we go. So does anybody have any questions about what relative fitness is? And again, if you're taking notes, you could just write down relative fitness equals or slightly equals survival of the fittest. And then maybe just put a little side note. Remember that survival of the fittest, not the most accurate term because it's more than just surviving. It's also about mating and passing along those genes that helped you survive. All right. Here's where it gets slightly more complicated. Not much. There'll definitely be questions about this on the exam. We're going to talk about the 3 general outcomes of natural selection. So when natural selection happens, if you're talking about the big picture, there's only 3 outcomes it could have. And for the exam, what I'm going to do is I'm going to give you some examples. I'm like, all right, you had a population of this kind of animal, and it changed in this way, right? I'm going to give you a hypothetical. And then you'll say, okay, well, which of those 3 outcomes was it? So with that in the back of your mind, let's talk about it. If we were to graph the coat color of a population of mice, we would get a bell-shaped curve. If natural selection favors certain colors over the others, the population will change. So again, well, I'm not even going to read this. It's there for you if you want to download the PowerPoint, but I'm going to use these terms as I show you the graph, because it'll make more sense if you see it in action rather than just reading the definition. And remember, for the exam, I'm not going to ask you the definition of directional selection or disruptive selection or stabilizing selection. I'm going to say this population changed in this way, and then you need to recognize that as either being directional, disruptive, or stabilizer. So here we go. Here's the original population. This is what I mean by bell curve, frequency of individuals. For those of you who don't know what this means, like a bell curve, what this means is a large majority of this population is this color, right? There's a lot more than any other color that is this color of mice. And then there's slightly fewer of this color, slightly fewer of this color, and even fewer of the really light ones and really dark ones, right? That's what a bell curve means. That's where the term curve comes from with a grade. Like in theory, the largest number of people in a class should have a C and should have even fewer D's and B's and really even fewer F's and A's. But anyway, there's the original population. Let's say something happens. They're living on an island. It's a volcanic island. There's a new a new eruption. So all the different stuff that used to be around, all the different colors are gone. And now it's basically a black island. So in that case, it would be very disadvantageous to be light colored, because then you'll stick out and you'll get eaten by predators. So because of that, over time, the population shifted. It went from being, now it's kind of more favored towards the dark colors, right? Or, it could have gone the other way. It doesn't have to go left or right. The point is it goes one way or the other. Not both, but it goes one way or the other, and that is directional selection. So if I give you an example on an exam of a population that changed and went to one of the extreme phenotypes, that is directional selection. I'll come back to the middle one. Let's talk about stabilizing selection. Let's say in this case, again, it's a volcanic island, right? So now everything's very dark, but then we have an ice age. So there's basically two colors on the island now. There's dark when the rock is exposed, and there's white where there's, snow and ice covering everything. So now all of a sudden it's really advantageous. Whoops, that's this one down here. Sorry. So now it's really advantageous to be really dark or really white. In that case, you have disruptive selection, right? So this gray color, you get less of that middle phenotype, and you start getting more of the extreme phenotypes, because now all of a sudden that's the advantage. So yeah, that's disruptive selection. And stabilizing selection would be the opposite of disruptive. Instead of getting rid of that middle phenotype, you would get more of it. And I don't know what would happen in this hypothetical scenario where being gray would be an advantage. But let's just say for some reason something happens and it's really bad to be really dark. It's really bad to be really light. So because of that, over time, you get fewer and fewer of the light and dark ones, and you get more and more of that middle color. And if we had more time, I could give you some more questions or examples of this. But I will share a video with you eventually, hopefully this weekend, that talks about this from somebody else. But are there any questions about the three different types of selection? All right, I know it's 8:50. But you guys can get off and just watch the video if you want. I just want to finish up sexual selection, because that's the end of the evolution. Sexual selection, you already know it anyway. It's easy. It's when individuals with certain traits are more likely to mate than others. So it's something about them, like a bright red colored cardinal is more likely to mate than the dark colored cardinal. Not because there's any advantage necessarily to being bright. It's just it's more likely to get laid, basically. And that brings us to a term called sexual dimorphism. And that's where there's obvious differences between males and females. Birds are a very good example of that, because, again, usually the male-colored birds are very brightly colored and the females are very dull. If you download the PowerPoint, you can watch a quick video about this subject. Your book gives some examples, like peacocks that have secondary sex structures, like this big old tan tail feathers that they have, which is great for mating because it gets the attention and the females love it. But it's really bad for surviving, because you're really a big target for tigers. And with a big old tail like that, it's hard to maneuver and get away from tigers. Anyway, that's that. There's some more pictures of some examples of sexual dimorphism, and also when a lot of times males mate to get or fight for mating rights. And then finally we get to the end, which we never talked about anyway, which is the evolution connection, and it's about antibiotic resistance, and I talked about that on Wednesday. So we are done. Woo! And the last word for attendance is penicillin, since it's right there. And that's just might be too hard for you to write. Let's do killing. Killing or kill is the last word for attendance. Oops. Any questions? All right. Well.
Hey, did you say like penicillin was one and then kill would be the last?
I decided not to do penicillin because I know that might be a hard one to spell.
Yeah, I was going to ask you how to spell it.
Yeah, so instead of penicillin, it is the word kill or killing, whatever, any version of the word kill.
Okay.
All right. Any other questions, anybody? We'll start chapter 19 when we get back on Monday. It wouldn't hurt to read over it this weekend because we have to go quick. All right. Well, I'll be online for office hours if you need me.
Have a good day.
You too.